Thank you for the introduction. Thank you, Julia, for asking me. Thank you, everyone who's listening. And I'm going to meet some more folks later today on Zoom chat, so I look forward to that too. And I have to say, I see my old friend Magnus on the screen as well. So hello, Magnus. Good to see you. So I'm going to talk today, as you can see from the title, about the evolutionary genomics of Vitis. And of course, everybody has an idea about Vitis because you either drink wine or eat table grapes, and those are all from the species Vitis vinifera. And I'm going to talk more broadly about the genus because it's a really interesting genus. And I'm going to talk about three topics. One, a little bit about domestication, but really it's more about structural variants and perhaps clonal propagation. I'm going to talk a little bit about sex determination because it's an interesting system from that perspective, and about patterns of introgression among wild species. And so you may ask, let's see, oh, please slides move, turn. They were turning fine before. No, of course they're not. There we go. You may wonder why I want to study Vitis at all. This is a fairly new system for me. And as was mentioned, I studied for years maize, and I understand I have the dubious distinction of following Jeff Rosabarz, who spoke last week. I know Jeff very well. He was a postdoc in my lab, and I know him well enough to know that he's far more interesting than I am. And so, again, it is a dubious distinction. But for years, I studied domestication in maize, and I was trying to understand sort of the dynamics of domestication. And it became apparent to me just only recently. I've quit studying domestication for many years, and then I realized that actually perennials like Vitis vinifera have very different dynamics. So one of the reasons that I wanted to look at Vitis was because of this interesting issue of perennial domestication. Now, I'm not going to talk much about it, but it's something to keep in mind. The other reason that I really think Vitis is a great system, an interesting system, is because as a genus, it's really remarkable. It has about 70 species, 35 of which are in China, roughly 35 in North America. There's only one European Vitis, and that's Vitis vinifera. But the wild species are incredibly important agronomically, and they're interesting evolutionarily as well. So agronomically, they're important because almost every Vitis vinifera, all viticulture is based on having rootstocks from North American wild species. So understanding these species is incredibly important for viticulture. And these species have incredible morphological and ecological diversity. Morphological diversity, I have here a picture from a recent publication, not my own. These are all North American Vitis species. You can see the leaves differ remarkably among these 8 species shown here. All of them, or most of them, are used as rootstocks, by the way. And there's great interest in this morphological diversity and what generates this morphological diversity, as well as ecological diversity. They're mostly found in stream beds, but they're found in very hot environments. I'll talk about some that grow in Arizona. They're found on serpentine soils. There's lots of specialization among these, and there's even some indication that there's been hybrid speciation. So from an evolutionary, ecological, morphological standpoint, there's some really exciting evolutionary questions. Another reason that I thought this would be a fun genus to study is because there is an existing community that really cares about this genus. Of course, the major part of that community is viticulture, representing billions and billions of dollars yearly, in wine grapes, etc. But there's a lot of physiology and a lot of interest in the ecology of these species. And so, it seemed like it's a great species to study for that reason. And another reason I wanted to study them is because they have relatively small genomes of about 500 megabases. Now, as you'll see in just a minute, these genomes, while small, and it seems like, wow, these should be so easy to study, they're not, because they're incredibly heterozygous and a bit of a mess. So it's actually a complex and challenging system to study from a genomic perspective. So when I thought, okay, Vitis seems to be the right system to ask some really interesting questions, the first thing I did is try to find some collaborators. Now, I'm in Southern California, but up in Northern California at UC Davis, where, by the way, Jeff Rosabarz is, they have a viticulture department. And the first person I reached out to was this fellow named Andy Walker. Now, Andy is the only great breeder in California. He has an amazingly well-supported research program. But one of the things that's really great about Andy, and in fact most breeders, is that he's actually quite the naturalist and he knows the various species quite well, Vitis species. And in fact, for 30 years, every summer, he takes a drive through the Southwest United States and samples accessions, brings them back to UC Davis, and he has this living collection of wild accessions from many species. And so I contacted Andy. He seemed excited to do something together, and he said, if we do something together, we have to include this fellow, Dario Cantu. If you don't know Dario, he's really, his sort of buzzword is the genomics of the vineyard. And he studies everything in the vineyard, from the grapes to the pathogens. For example, here's a picture of botrytis, the noble rot that makes really fancy wine, and he's sequenced the genome of that. So everything that I'm going to tell you about today is in collaboration with these two groups, and I just wanted to make sure you knew about them right from the beginning. So I'm going to talk about 3 different topics. I'll try to watch my time carefully. I tend to talk too long. These 3 different topics are the following. The first is I'm going to talk a little bit about genomics, a little bit about domestication, but really more, I think, about clonal propagation and the effects on structural variation. Then I'm going to talk about the sex determination locus. This is a really interesting feature of Vitis because it's a dioecis. In the wild, they are dioecis. They have separate male and female individuals. In cultivation, they're hermaphroditic. So there's been some interesting changes that have occurred in sex determination. And finally, I'm going to talk a little bit about adaptive introgression among species. Let's start with the first topic and touch a little bit on domestication of Vitis vinifera. So Vitis vinifera was domesticated about 8,000 years ago. We don't know, right? These are always fuzzy numbers. And we're not even sure if it was domesticated once or more than once, but at least there was one major domestication event in Georgia, near Turkey. There was associated with domestication a moderate, in fact, almost undetectable genetic bottleneck. And this gets a little bit at this point about perennial plants versus annual plants. Most annual plants show this great restriction in genetic diversity after cultivation. It's part of the domestication process. Perennials typically don't show this for various reasons. And again, I'm not going to talk about it much today, but I think it's a really interesting feature of domestication. But like all domesticated species, it's undergone some phenotypic shifts, changes in leaf size. I told you a little bit about under domestication. There's perfect flowers. They have both male and female parts. But in the wild, all the wild species have separate male and female individuals with separate male and female flowers. Changes in things like the size of grapes and the number of grapes in a bunch, and changes in the seeds. So when we set out to start studying Vitis, we thought, let's just start modestly and do something that no one had done. And that's just sort of do a whole genome resequencing study and, include some wild grapes, Vitis vinifera sylvestris, or the wild grapes, and the cultivated grapes and wine and table grapes, and sort of just do a population genetic study. And I don't want to go into details about what we did, but I do want to glean a couple of interesting features from it. Oh, by the way, all this work that I'll tell you about in this first section was by a really talented postdoc who now has a professorship in Shenzhen, Yongfeng Zhu. So we did some modest resequencing, and we did, the typical things you might do with a resequencing study. You do things like compare genetic diversity between wild species, or the wild accessions, and the cultivated accessions. And here you can see they're about the same level. This goes at this idea that there really wasn't a strong genetic bottleneck at the time of domestication, or subsequently, something happened where you don't see this genetic bottleneck. But we did notice something that was kind of interesting, and that is that heterozygosity within the cultivated accessions was quite a bit higher than with the wild accessions. Okay, so maybe that's puzzling, maybe it's not so puzzling, but what became clear to us is that this is a system in which you have clonal propagation. Some of these varieties, cultivated varieties, go back to Roman times. They've been clonally propagated for thousands of years. And what happens is when you have this situation of clonal propagation, you have the opportunity to hide deleterious recessive SNPs. So you can get deleterious changes, and these can be things like amino acid changes that are not conservative or even stop stop mutations that cause a premature stop codon. And what we found is that when you measure these kinds of mutations, these deleterious or predicted deleterious mutations, the cultivated accessions had many more than the wild accessions. Now that's not surprising. This is called the cost of domestication. You see this a lot in domesticated crops. But the heterozygous load was in fact very, very high. And so most of the deleterious SNPs were found in heterozygotes. And again, intuitively, that's easy to understand. You have clonal propagation. If you have a recessive mutation that's never going to be homozygous, then it's really, it's shielded. And we sort of messed around with some simulation methods to kind of look at the dynamics of this. And it's as you might intuit, except for a couple of really interesting things. So what these are simulations that go forward in time. And they have various features. For example, some of them are outcrossing populations. So here we have an outcrossing population. Here we have an outcrossing population. And that outcrossing, some of them have recessive alleles, deleterious alleles, and some of them have additive deleterious alleles. And what you see with outcrossing is that fitness really doesn't change. Outcrossing does a good job sort of balancing the deleterious load and getting rid of the deleterious load. What happens when you become clonally propagated, however, if you have additive deleterious variants, your fitness goes down in a hurry because it shows up as a heterozygous, it affects fitness, you can't get rid of it because there's no recombination, boom, you're in trouble. But what's kind of interesting about when you clonally propagate something is that actually you get this immediate increase in fitness when there are recessive deleterious mutations. And the reason is, you get this increase in fitness is because they're completely hidden and you're not getting any deleterious load in the homozygous state. And so what actually happens is your fitness remarkably increases. This is all just to say that there's good reasons to expect that you get this accumulation of recessive deleterious SNPs in clonally propagated plants. Fine. We've seen this with SNPs, but what about structural variants? Because structural variants, which are big changes in genomes, inversions, deletions, insertions, those kinds of things, are they also accumulating in these clonally propagated plants? So we wanted to ask that question, and to do that, we sequenced and assembled the Chardonnay genome using PacBio sequencing. So long read sequencing where you can really look at big chunks of the genome. Blah, 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 right? It's a very nice genome, N50 of 24 megabases, and it has about 38,000 genes, okay? Great, so we can make a genome, and it actually was a little more complex than you think because of the incredible heterozygosity in grapes. But one thing we found was that there's actually a ton of genic hemizygosity. That is, there's a lot of gain and loss of alleles for genes. So here I'm showing you a track of sequencing along part of a chromosome 2. I have here genes, and you can see one of these genes is incredibly heterozygous, and you can see that because of the colors, right? Those are heterozygous SNPs within that gene. And you can also see that this gene has about double the coverage of the genes next to it. That's because the genes next to it are homozygous. Okay, and so I once asked my Drosophila colleagues down the hall, I said, how homozygous do you think Drosophila could be for gene copies? And they said, no more than a couple percent at most are homozygous. And I said, well, would you believe that 1 in 7 genes is homozygous in Chardonnay? They said, no, they don't believe it. And I'm not sure we did either. And what we did, and actually to estimate how many genes were homozygous, because we had long read sequencing, we actually required positive information about hemizygosity. That is, we required that we had a read that mapped on either side of the gene, but lacked the gene itself, right? So it's pretty strict, but we still thought maybe we're completely wrong. So we did this comparing Chardonnay reads to the Chardonnay reference. We got about 15% of genes are hemizygous. They're only one copy. They don't have 2 alleles. We did this mapping Chardonnay reads to another reference. We got 15%. We did Cabernet reads, this is from Dario's labs, to a Cabernet reference, we got 15%. We now have a Zinfandel genome, we did that, and it's less heterozygous both for SNPs and for hemizygosity. And so the point is that you can summarize it however you want, but around 1 in 10 genes is hemizygous. There's only one copy of alleles. Now, this down here represents rice, and we thought, listen, maybe our method's completely wrong, even though actually we all used different methods when we were doing this. And so we said, why don't we use a null negative control? And so we compared rice to rice, long reads of rice to the long read rice reference using the same approach. And we got about 0.7% gene hemizygosity. So I think this is probably our error rate. Remember, rice is selfing. It's got to be hemizygous or homozygous by definition. And if you look at a read, you know, the reads of rice, you don't see these shifts in coverage or shifts in or even heterozygosity as you expect. So maybe the error rate of the method's about 1%, but that still means that, you know, roughly at least 10% of genes are hemizygous, which is pretty crazy, unexpected to me anyway. So the next question is, do any of these structural variants have any effect on phenotype? And we ran across this really interesting story, I think, about a large inversion on chromosome 2. So what I'm showing you here is Pinot Noir versus Cabernet. And you can look at chromosome 2 and you can see that they're pretty linear, though there's some differences, you know, insertion, deletions, probably transposable elements that differentiate them. Now look what happens when you look at Chardonnay. There's this big inversion on chromosome 2, and you say, big deal. Inversions happen all the time. But it's where this inversion is that's quite interesting, because this inversion is about 5 megabases long. It has 176 genes, and here's the key thing. It includes the MIB genes that affect the color of berries. And you can see Pinot Noir and Chardonnay have very different color berries. So berry color is one of these really cool phenotypes in plant biology that we know about, that we know the genetic cause. There are 2 MIB genes that affect the accumulation of anthocyanin to make dark grapes. One of those is knocked out in white grapes because of this transposable element in front of the gene, and it stops transcription. The other gene is something called MIB2, and MIB2 has a null mutation, and you need both of these mutations to make white grapes. You need to knock out both of these genes. One of the things that's very cool about grapes is these 2 null mutations are heterozygous in many of the dark buried lines. So in other words, the dark buried lines are carrying these recessive null mutations, and they have the potential to become white grapes if somehow you make the null mutations homozygous. So how does that happen? How do you make the null mutations homozygous so that you make white grapes? Well, turns out they don't become homozygous, they become hemizygous. Let me show you how we know that. We know that because of a paper not from my group that I think it's one of my favorite papers. This is the Tempranillo grape. For those of you who like wine from Spain, Tempranillo, it's my favorite variety. It's usually red, tinto, but this is a somatic mutation that caused white grapes on this particular variety. And what is the somatic mutation that occurred? Well, it turned out on chromosome 2, there was probably an inversion. They're not sure about that. And there was a reduction, it became hemizygous in the region of the MIB genes. How do they know it became hemizygous? What they did is they mapped the read depth of the white grape versus the red grape. And as you go along, you can go blah, 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 and suddenly the coverage in the white grape is much lower. There's still coverage of this region, but it's hemizygous in this entire region. Okay, so we also see for Chardonnay an inversion. It has to be a different event because this is a somatic mutation in the field. And the question is, was our inversion hemizygous? Did it lead to hemizygosity? Well, here's another one of these read length things that I showed you before. And you can see coming up on chromosome 2, coming up to the version, you see lots of colors. It's It's not homozygous, it's heterozygous, but it's not hemizygous. Suddenly you get to the inversion, boom, it's hemizygous. And it's hemizygous all the way through to the end. So our inversion somehow caused hemizygosity, and that hemizygosity made the null mutations, which are typically recessive. They've knocked out the MIB genes, and now we have a white grape. So then the question becomes, is this always what happens? That is, when you get a white grape, or a green grape, whatever you want to call it, is it always due to this hemizygosity and a large genomic rearrangement? So far we have an N of 2, Chardonnay and Tempranillo. And what we did is we studied, we took as many sequences as we could from white and dark grapes, and you can see from this phylogeny that white grapes have shown up many times throughout the history of Vitis. And the question is in every case, is there some evidence that these null mutations became hemizygous and that there's some sort of inversion or rearrangement? And I won't go into the details of how we did this. We only had short read sequences and not the sort of now industry standard long read sequences. But we made comparisons between white and dark grapes, the genomes, with whole genome resequencing. And the take home was, in every case, we saw this sort of loss of coverage in the white grapes, meaning they're hemizygous in that region. And in every case, we could find some reads that suggested there had been an inversion. So when we get white grapes, and I think this is a great story because this is one of the things that has been solved in plant biology, really one of the very best known phenotypic traits that we know the genetic causes of them. But what we didn't know is that when you get the somatic mutations to make white grapes, they're often due to inversions that make the whole region hemizygous. So we were able to invert inversions on chromosome 2, and I think 6 comparisons. Those inversions changed in size. That makes sense. They're not always the exact same inversion because they happened on several different occasions. They included from 134 to 176 genes, 118 genes in common. We looked at those genes. We didn't see anything really interesting. We think it's really all about the MIB genes. Whoop. So that's what I want you to get from the first part of this talk. That clonal propagation provides an opportunity to hide recessive deleterious variants. And that really, I think, is what leads to the very high heterozygosity and hemizygosity in cultivated grapes. So we have 1 in 7 genes are hemizygous in Chardonnay. That's our best estimate. We still would like to refine those estimates, but we think that's about right. We found large mega-base scale inversions that drive berry color evolution. And I think this is a really interesting and dynamic story about how phenotypes can change due to very unexpected genetic events. Again, this is probably one of the best known phenotypes in plant biology, but what had been missing was the fact that often this phenotype is being facilitated by these large scale chromosomal events. Okay, so that's the first part of my talk. And now I'm going to talk about something really quite different, but hopefully interesting to you. And that's the sex determination locus. So when we did our first studies, first with whole genome resequencing and then actually looking at structural variants across the genome, one of the standard things you do when you're studying domesticates is you compare the domesticate to its wild relatives. And you do things like FST, or you look for diverged regions of the genome. So here, when you look at SNPs, you can see we go along this Manhattan plot. There's 19 chromosomes, and you can see there's this peak of divergence right here on chromosome 2. When we look at structural variants, and I haven't really talked about how we looked at those, but we can do the same thing, and you find slightly different peaks. One of the peaks is the berry color. I just told you about that peak because there's this huge inversion in that region that happens repeatedly. But also, you also see this peak in the sex determination region on chromosome 2. So what is this region? It's clearly very different between domesticates and wilds, and what's going on in that region. Well, you'll recall that I told you that all the wild species, these species go back about 30 million years, by the way, they are dioecious, meaning they have separate male and female flowers. During domestication, there was actually a shift in mating system so that the flowers are now hermaphroditic. They're perfect flowers. And so there's been this remarkable shift during domestication. And this has happened a couple of times. Grapes are not unique, and I'm struggling to remember the other species in which this happened. Sorry, I just had a brain blip. But it's rare, but it's not unprecedented, is my point. Okay. The thing that you need to know is that we know something about the genotypes of this of sex determination. Females are homozygous females. That's what the F is for. Males are heterozygous males, females. And so it's a little bit like, frankly, like humans, you know, XY and XX. The hermaphrodites can be either heterozygous, hermaphrodite female, or they can be homozygous for hermaphrodites. And when I say this, when I say, for example, that it's like humans, XY and XX, I do want you to keep in mind, however, that there are not separate sex chromosomes in Vitis. There's, in fact, a small region on chromosome 2 that determines sex, and that region had been mapped previously. So before I start talking too much more, I have to tell you a little bit about this long history of thinking about how sex changes in the plant world, sex mating systems. And this all goes back, really, it's by a model made famous by Charles and Charlesworth, who really was first put forward by Westergaard generally, but actually remarkably, even earlier in the Vitis world in 1938, there was some talk about how sex determination occurs. Okay, so in the plant world, it's thought that being a hermaphrodite is ancestral. So here we have, you know, a nice perfect flower. And that in order to get to two separate sexes, to evolve two separate sexes, a couple of things have to happen. First, there's a male sterility mutation that leads to males. Once it's homozygous, it's recessive to start with. And then there's a second female sterility mutation that leads to males. Sorry, I had this back. This is males, females. Hopefully you're not too confused, but the main point is there are two mutations, and there's been a lot of study of different plant systems trying to find these two mutations. Once you have these two mutations, the question is, how do you become a hermaphrodite again? And it's hypothesized you need a recombination event between the male and the female sterility mutations. So you get this recombination event that could cross with the female, for example, and you can get sterile flowers and or perfect flowers, okay? So there's this long history of this model, and I wouldn't worry too much about understanding this model, but I would ask that you recognize there's a few really interesting questions here. One is, what is the recessive male sterility mutation? The second is, what is the dominant female sterility mutation? And is there molecular evidence for a recombination event that would lead to reversion back to being a hermaphrodite? And if so, remember that the genus has had dioecy for about 30 million years. And why hasn't that system broken down? If it can break down in cultivation, why isn't it broken down in nature? Why hasn't there been these recombination events that lead to hermaphrodites? So we tried to approach this problem by first looking at this region of the chromosome, because for the first time ever with our Chardonnay genome, we were actually able to resolve very carefully this whole region. And I don't want to say too much about this, except to say you're going to see another slide like this that shows the whole region. The genes in the region are the arrows, and I'll talk about some specific genes, but don't worry about too much about these. Chardonnay, we actually sequenced because we knew it had two hermaphroditic haplotypes at this locus. Dario's group also sequenced Cabernet Sauvignon, which was a heterozygous for hermaphrodite and females. And what we were hoping is that this data would give us some insights into what are the male sterility mutations and what are the female sterility mutations. And this was work done by one of Dario's postdocs at UC Davis. And You know, she did a wonderful job annotating the region, being very carefully doing it all by hand. And the truth is, though, that we just didn't have the right data set to figure out some of these questions because we didn't have any sequence information for males. So what we did as a group is we went out and sequenced a bunch more genomes, including wild species from males. So we sequenced these all with long read PacBio sequencing, another species from Arizonica, and an outgroup, rotundifolia. And by the way, I've been saying it's 30 million years. It's more like 20 million years that the dioecious system has been in existence. And the point I want you to know is that we got all this data, and we localized from each genome both haplotypes using sex-linked markers. And we inferred male, female, hermaphrodite haplotypes. And now we can actually compare among them with a pretty good sample of 22 distinct haplotypes. Remember, each of these haplotypes is, well, it's about half a megabase or so. Okay, so now we have all these haplotypes and we can start looking at gene content in the hope that we can find something about these male sterility or female sterility mutations. Again, I'm not going to go into the details of this, but there are a few things you can see right away. Here's a male haplotype from a wild plant. Here's a female haplotype, and you can see there's big differences in length. And that seems to be consistent across the different male and female haplotypes. The hermaphrodite haplotypes are also long. You can do silly things like this and compare the Cabernet Sauvignon H haplotype to other hermaphroditic haplotypes, and they're all about the same length. If you look at the hermaphroditic haplotype from Cabernet Sauvignon versus females, female haplotypes are shorter. And if you look at the male haplotypes, they tend to be longer, quite a bit longer than the hermaphrodite haplotype. And interestingly enough, in one of these genomes, there's a large inversion in this section of the genome, the sex determination region. And that's actually kind of interesting because remember, one of the questions that we were interested in is, why hasn't this system broken down? If all you need is recombination between male and female haplotypes to recreate a hermaphrodite, why hasn't it broken down? Well, in this one species, our outgroup species, it's probably because there's an inversion and that inversion blocks recombination. Okay, so remember now, we have this 22 haplotypes, very nicely resolved, done by hand, beautiful work by Melanie. And now you can do some silly, simple things. For example, what if you look at nucleotides that are completely male specific or completely female specific, and you map the incidence of those nucleotides along a hermaphroditic, the Cabernet Sauvignon haplotype? What you see is that there's a region that tends to be male linked, and there's a region that tends to be female linked. Okay, cool. What does that mean? That means our hermaphroditic reference had a recombination event somewhere in here. Great, we've already learned something important. Yes, there's evidence for recombination. That's why it went from dioecious to becoming hermaphroditic again. Cool. But what else this tells us is that the female sterility mutation must be somewhere here in the male part, and the male sterility mutation must be somewhere here in the female part of this section of the genome that, again, is about 0.3 or 4 megabases, less than 0.5. So we looked at all the different genes within the male region, trying to find the female sterility mutation, and one particular gene knocked us off, and we think we know what it is. This particular gene is called the YABI gene, and it's clearly the male haplotypes are very different than the female haplotypes and the hermaphroditic haplotypes. That's what you'd expect. So in this phylogeny, it's highly expressed in male flowers, so we looked at gene expression for this region as well, and actually generated developmental series of flower development. What's really interesting about YABI is that in Arabidopsis and maize, these genes are involved in floral development, especially the ovule. And we know that overexpression of YABI in Arabidopsis and in orchids leads to abnormal female development. So there's ongoing work actually trying to confirm this inference So far, all the evidence is pretty strong that it looks like this is, in fact, the female sterility gene. Oh, and here's just some Arabidopsis photos. We don't have to worry about those. So we think we found a female sterility candidate. But what else is going on here? Here's the male region. Here's the female region. And now what if we looked at sex-linked indels? And these are small insertion deletion events. Again, they show that there's a male region of the hermaphrodite, the hermaphroditic haplotype from Cabernet Sauvignon. There's a female version. And what we did is we looked at these indels to see if there are any genes that seem kind of obvious as potentially the male sterility mutants. And we found that there is a couple of genes that have premature stop codons in the female part. And those premature stop codons, the one that's really knocked our socks off, is this indel event that has been maintained for 20 million years, as long as the Diaceae, in this one gene called INP1. So here is just a lineup of females and then males. And there's this indel event that leads to a premature stop codon in females. And so we think knocking out this gene causes males. We looked at F1 populations and all this stuff, and it segregates perfectly with sex, but that's maybe not such a big deal because this region has very low recombination and segregates perfectly with sex anyway. But this gene is quite interesting because in Arabidopsis, if you mutate this gene, it causes problems with pollen. And so it does seem to be a gene that's really a highly important candidate for causing male sterility. This is, I think, is that from Arabidopsis? I don't recall. But the important point is we think we have this gene, and actually Dario is engineering this knockout and testing if this is really the male sterility mutation. Okay, so what did we learn from the sex determination locus? I'll say again that the whole question of mating system is a very important one in plant biology. It's been studied in several different species now. In few cases do we really know what's exactly in a sex determination locus, and there's lots of reasons for that. So we now really have studied this region in great detail in grapes. And we think we've found the recessive male sterility mutation. It needs to be confirmed functionally. It's this thing called INP1 that actually has to do with pollen development. We think we found the dominant female sterility mutation. It's this thing called YABI, which if you've overexpressed, it messes up female development. Is there evidence leading to a reversion? Remember that cultivars are hermaphroditic, and do we see evidence of a recombination event? Yes, absolutely. We know where that recombination event is. And I will say, it looks like maybe this has happened more than once. So there may be several recombination events that occurred during the cultivation or domestication. But then that leads to the question, if all you have to do is recombine to break down this whole Diaceae system, why has it been around for 20 million years? And what's keeping these recombination from occurring? Well, I showed you one case where there's an inversion, which probably is a reasonable hypothesis to think that stops recombination. But in general, we don't know why there has not been more recombination leading back to hermaphrodites in nature in Vitis vinifera. Though we think, my guess is that the different lengths of the haplotypes, males are much longer than females. They're filled with transposable elements, which maybe might be methylated. We don't know yet. They might retard recombination events. And so maybe there's this system that's built up, not unlike other systems where recombination has been suppressed. Okay, I think I'll have to go fast on this last section. Thank you for being patient with me. Just to remind you, the first thing we learned about was really about hemizygosity and the effects it can have on one phenotype, which is grape color. The second, we're talking about another phenotype, which is sex determination, and we think we found some candidate genes that are being confirmed for that. And now I want to talk about actually another phenotype, and we didn't start out really to talk about phenotypes. We just wanted to answer a simple question. And that is there adaptive introgression between Southwest species? Now, why is that an interesting question? Adaptive introgression is now all the rage these days due to studies like this one. So this is a study in Science in which they were looking at different groups of heliconius butterflies. These are, of course, these highly patterned butterflies that the pattern is also very adaptive. It has to do with, predation and all these other things. And what they did in this study, which it's a great paper, by the way, is they sequenced a bunch of genomes, and then they actually, made a phylogeny tool. That's the dark black part. That's the consensus phylogeny. But then they found regions of the genome that seemed not to fit the phylogeny. Now, you can have regions of a genome not fit the phylogeny because of lineage sorting, right? Or just phylogenetic lack of resolution. But they went to great lengths to show that some of these things really appear to be due to introgression, not lineage sorting, but actual introgression. And really what's remarkable is that the introgressed pieces often contained genes that contributed to color pattern. Okay, so why is that important? First, typically when we think about gene flow or gene exchange between, say, in this case, two species, you'd expect it to homogenize the species. But phenotypically, what it's actually doing by exchanging these pattern genes is it's actually allowing new adaptation and patterns, new patterns, novelty, genetic and adaptive novelty. And it's just really, I think, becoming appreciated in the last maybe 5 years about that introgression can do this and cause and contribute to novelty, ecological, evolutionary divergence. Another really great example, the figures from this commentary that I and Yongfeng wrote, but it really comes from a group from Lauren Riesberg's lab, this paper. And Lauren has always studied sunflowers, and he found that there were really interesting cases of introgression. For example, an introgressed region of the genome that seems to allow adaptation to growing on sand dunes. So this ability to grow in sand dunes occurred once, and then it got introgressed between species. So now that multiple species can grow in sand dunes. My point is that adaptive introgression is really fascinating. It's being more and more appreciated for its role in contributing to divergence among species, and especially adaptive divergence. So we are, as you know, interested in the North American species of Vitis because they're so important for rootstocks. And we were interested in knowing, has there been introgression among them, and has it been common? If so, to what extent of the genome is represented in these introgression events? And what are the evolutionary processes that drive this introgression? If so, what traits are affected? So what we did is I'll say again, Vitis is a great system for a lot of things. And one of the things it's great for is looking at these kinds of adaptive introgression events. Here's why. They're all interfertile. So there's roughly 35 species in North America, and they're all interfertile. They are often overlapping geographically. And so this map shows you how many taxa are in different parts of the North America. I guess that's all I want to say, that these rootstocks are sources of biotic and abiotic resistance in cultivation. And one of the things we're really interested in as a group is Pierce's disease, which is a relatively new disease that's now affecting olives in Europe as well. So it's quite pernicious. So what we did in this study is we went to Andy Walker's collection from the Southwest, and we sequenced about 130 genomes using short read technologies, the old school stuff. We looked at 6 species, and the idea was we wanted to get sort of this population genetic sample that we could start to look at introgression. And this was work done by a postdoc in my lab, Abraham, and Honas, another postdoc in the lab, who teamed very nicely together. So we sequenced about 130 such samples. In the end, we found that some were hybrids using, structure-based approaches. So in the end, we had to get rid of about 19 of our samples, and we had 111 samples. And our question is, can we find historical introgression between species in these samples? Just as sort of a visual of the data, we could, create consensus trees with all our accessions using SNPs from throughout the genome. The species group very nicely together, but these red clouds behind them are separate genes that are being applied for phylogeny, and there's a lot of lack of resolution. There's probably a lot of lineage sorting going on here, and the question is, can we actually find regions that are introgressed? Sorry, I'm running through this because I'm running out of time, but we did just very basic sort of analyses, and these are the famous ABA-BABA tests in which you use 4 species. And in these tests, you have a control species, a species that's a receptor, that is, it receives genetic variants from the donor. So this is where introgression occurs. And then you have this outgroup that you need to sort of polarize the test. And what we did is we just used this basic setup with all of our 6 species. And so we looked at all the possible combinations of species that we call trios. And we looked at all the combinations of species that make sense phylogenetically. And we asked, is there introgression between them? So for example, here's one of our trios. Here's our outgroup. Here's something called candykins. Here's something called Riparia. Here's something called Arizonica. And with this trio, we can ask, has there been introgression from candykins into Riparia? And we applied this thing called a D statistic. And this is kind of the basic results that we found. Very briefly, we found that Berlandiaria, V. Burr, there's evidence that pieces of V. Burr went into Vitis riparia, and that about 8% or 7% of the genome of Vitis riparia can be attributed to introgression from Berlandieri. So about 8% of the genome, 7.5%. There's 2 numbers here because we had 2 different control species. But the point is, here we have, went the wrong way. Now it's going the wrong way again. The point is that we have these 6 species that we were studying, and that most of them showed some evidence for introgression between them in kind of a complex pattern where one was donating to 2 different species, candykins to Berlandieri, candykins to Riparia. Riparia seemed to be a receptor species from 3 other species, but then also was donating to another one, Gerdaniana, and so it's completely complex. Now, one of the things that we found really interesting about this is that most of the species that we see with an arrow between them showing that there's introgression, or we're inferring that there's introgression, they actually in the present don't grow in overlapping distributions. And so what we did is species distribution modeling, where you can sort of use bioclimatic variables to estimate where species lived in the past. And what we found is stuff like this, this is 2 species, candykins and Riparia. So here we see candykins seems to have contributed to Riparia. About 2% of the Riparia genome comes from candykins. And what we found is that they overlapped much more in the past, using sort of climate modeling and climate prediction modeling than they do in the present. And that we found that to be true for all of these species. And so it suggests that most of this introgression that we're seeing is really historical and not recent introgression. You can then actually say, where are the introgressed regions of the genome? So here's all the 19 chromosomes. There's a method called FDM, where you can look at peaks, and the peaks represent potentially introgressed regions. So here I show little regions of introgression in red. Again, this is all inferential. There's no proof, it's evolutionary inference. And we called all these red regions putatively introgressed regions. So what do we learn about these introgressed regions? Some basic things. If you look across all the species and all these introgressed regions, they tend to be gene poor. Now, that's actually common in the introgression literature. Most introgressed regions are gene poor. They don't have a lot of genes, perhaps, well, we can go into that later. But even though they are gene poor overall, they are enriched, all of these regions, or most of them, for disease resistance genes. So this is giving us a clue about maybe some adaptive introgression. If you get these disease resistance genes moved among species, then maybe you're having your adaptation to biotic stresses that are moving between species. So I won't go into detail in this, but essentially the arrows, it's the same figure you saw showing the different species with arrows showing introgressed regions. Where the arrows are green, that means there's really a significant enrichment of disease resistance genes within these introgressed regions. Here's just an example. Again, I'll run through this fast, but here's these introgressed regions, and some of them, the putative resistance genes are in green, the other genes are darker, and in some cases, it's like 50% of these regions are made up of resistance genes, when the genomic content is about 2% of the genome is these resistance genes. So they really are enriched in gene clusters for disease resistance. So we thought, well, if that's true, then maybe they should be associated with some sort of phenotype that has to do with biotic resistance. And we're interested in Pierce's disease for lots of reasons. It's a horrible pest for the viticulture world. And so we did these assays in which you actually take this bacterium and you put it within plants in the greenhouse, and then you measure later how many bacteria are in the plants. And essentially, we just wanted to find out if we could find associated SNPs first with this large panel of 130 or so accessions. Second, are those SNPs enriched within introgressed regions? Suggesting again that these introgressed regions play a role in sort of moving biotic resistance across species. This was work done by Samara at UC Davis, and I'll just cut to the point. If you looked at the introgressed regions in some of these things that we call trios, some of them are highly enriched in associated SNPs with Pierce's disease. Others are highly enriched in disease defense genes. I'm not showing that statistic here, but I'm showing you a line there. And so in most cases, these introgressed regions either are implicated in function in biotic resistance to Pierce's disease or have these defense disease regions. So they seem to be associated with disease. They are also associated, enriched for associations with bioclimatic variables. So you can go to worldclim.org, you can get all this bioclimatic data. I won't say too much more about the bioclimatic data. You can look at bioclimatic data and put them on maps and do fancy things. But the take home is we could associate bioclimatic data using various approaches. And here's what I really want you to see. Again, I'm going through this very quickly, but I want you to see that there's really significant enrichment, more SNPs associated with bioclimatic variables in the introgressed regions than in the rest of the genome, okay? And so it seems like there is a signal if A, we have the right introgressed regions, and B, we've done our associations properly, there's a strong signal that these introgressed regions are really enriched for adaptation to biotic and abiotic stresses. This fits like with this idea about butterflies that they're moving around cassettes for adaptation, and we think we're seeing very similar dynamics on a different scale in Vitis. So to summarize, the introgressed regions compose from 2 to 8% of extant genomes. There's some kind of problems in the way we did this, but we do know that they seem to be enriched for disease resistance and bioclimatic variables, and overall it suggests introgression fuels abiotic and biotic adaptation. So, sorry, I'm going on too long. That's our third section. And hopefully you can see that there's some what I really want you to take home from this is that there's some really interesting things in this genus. It seems I'm having a great time working on these problems, and hopefully at least one of these stories interested you. Let me give you my acknowledgements. My lab, I think that was it looks like the Last Supper, and I think it was the last lab lunch we had before we're all quarantined, so it really was the Last Supper. The Cantu lab, which is Dario's just an amazing genomicist and biologist. He's fantastic. The Walker lab with Andy and Sumera, and then our funding from various sources. So again, talked a little too long, but happy to have questions if there's time.